I wish to ask now our first speaker, Dr. Miomir Kneževič, to present the lecture. He is a biologist specializing in biotechnology with more than 20 years of experience in gene and cell therapy, regenerative medicine, and molecular biology. He is a former university professor and currently course scientific position director at GaiaCell. I'm very happy to have him here.
Thank you.
Thank you.
Thank you very much for the invitation and to have an opening lecture of the conference. And I hope that the material which I will present will help us to, let's say, start or have a better start in our longevity projects in the future. So as Mr. Lipošek said, I'm from the startup company GaiaCell for advanced cell and gene therapy, where we are developing new techniques and technologies and products for cell and gene therapy, as it said. This cell and gene therapy is part of the so-called advanced therapy medicinal products regulated in Europe, which consists of four major groups or pillars. One is somatic cell therapy, then tissue engineering, combinatorial therapy, and gene therapy. So it's quite diverse, and it's regulated relatively strongly, and it's hard to achieve all the standards which are necessary. So I would like to, in this relatively short period, just touch different aspects which are coming from technological, basic one, to the accessibility and use of these products and cells now and in the future, and some challenges which we are facing. I use, of course, some material which you will see here from different web pages. So first is what are the signs of aging? We know almost all of them. We see one is impaired vision, then impaired hearing, and then, of course, muscle strength, then bone density, and what is important very much is decreased immunity system functions, which are a source of a lot of problems in secondary stage. After that, let's say we can see also cognitive dysfunctions, and with this combination with depression and worse psychological health. So logically is what is the can we cure or can we at least stop aging, or let's say at least have it better acceptable for us? It is still in development, of course. Our body consists not of drugs, of tablets, but consists of cells. And logically is that during the development and our lifespan, our cells are dying. They are then dividing, differentiating, and more than 200 different cell types are there, showing all the possible combinations to let's say, to allow us to live and maintain our life properly. So the damage which can come from the injury or which comes just from living, it means from aging, is something which is necessary to touch when we would like to get better results with aging. So, of course, to use stem cells for anti-aging properties. Now it's still, let's say, more or less proved on animal models. On systematic research on humans is still missing. There are a lot of clinical trials which you can find in different databases, especially clinicaltrials.gov, and they are linked with aging frailty, where they show positive results which last several years. There is no real cure for aging. This we have to be sure. So the thing is that what we would like to do is let's say, we are satisfied if we say, if it is as it is, and I feel okay, then I would like to keep it as long as possible. And as it was mentioned before, when the lifespan is, let's say, prolonging, then we at the end will definitively, let's say, we want to die healthy, I would say, and feel well. If we just want to prolong, let's say, life which is not of higher quality, this is not what we really want. So we have to work on different levels. One is use the potency of the cells as they are, then change the cells ex vivo, it means in the laboratory, manufacture them, treat them with certain substances and so on, and later then inject or implant in the body to restore the functions, or let's say use genetic changes like gene editing and all new techniques which are allowing us to make cells, let's say, better. But this is, let's say, of course, the future. So what is the stem cell rejuvenation? With this term, we are using it to show the effects of natural aging process. So it means we would like to repeat it, and we would like to make it reproducible, and also, of course, first safe, then effective, and that we know what we can expect with such a treatment. So the thing is that in certain cases, it's also necessary that we get rid of the old or senescent cells, because it's known now that they are giving bad signals to the others, and somehow, let's say, put our cell traffic in the wrong way. And this is then not promoting the repair and regeneration. So if we cannot cure aging, so it means if we, but definitions say that this is an, that, let's say, aging is a disease, what is not, or it's a matter of the debate. We want to at least reverse this process and stop it for a while and make it better. These reverse aging technologies are, let's say, combination of different, not just stem cell products, of course, or stem cells itself, but also a lot of changes in lifestyle, which I think we will also hear a lot today, and different, let's say, substitutes and so on, which have to be balanced. But then, you know, all these recipes are here around, and there is no definite answer to it. So, and now the question, how to make it fair for everybody. Most of these so-called anti-aging or longevity therapies are now, let's say, exposed to public, let's say, debate about fairness of using these therapies. And in certain cases, they say they have to be accessible to everybody, which is, let's say, hard to achieve. Then they say these are not proven technologies, so insurance companies will not pay for them. Then the other thing is that in different parts of the world, let's say, such therapies can be then, let's say, easier or harder accepted. Also, there are ethical questions about it and sociological questions. And also, let's say, how fast can new technologies enter into the daily practice? And the question too, can somebody who would like to risk it or would like to pay for certain such therapy, is he or she allowed to do it? And if medical doctors are allowed to make it, and regulations here are now quite critical, and the acceptance in different parts of the world, let's say especially Asia and then Western world, are very different. Europe is highly regulated in this, and a lot of such therapies are still not available if they are not part of the clinical trial. But if we have the technologies which must be proved, of course, according to our rules, then it's also not fair not to, let's say, prevent them to enter in the certain trial, even if it is not supported by the big pharma industry. And we had now in one conference of Festival of Biologic, it was also the question about accessibility of gene therapies, because if you are using cell therapies also for very, let's say, I would say, life-threatening diseases or diseases which are, let's say, like spinal cord injury and so on, they must be, they are expensive, and who is paying for them? And they are, of course, most of them not proven completely. So the main thing is that here you have two pictures of the one injury of the embryo and the other one injury of the adult person. Adult person is a person after birth. Before you have embryo, fetus, that's a part where, let's say, on cellular level, things are very different. And what we would like to achieve, we would like to achieve regeneration as it is in embryo, also in an adult person. Why? The parts which are, let's say, result of the injury is first inflammation after acute injury, then regeneration, and the result at the end is also fibrosis. In adult person, acute injury, after acute injury, inflammation is much stronger, and it is also correct that it is, because it is also saving us from the infections, and it's also helping the whole process. But the problem is, if the inflammation is too strong, then instead of regeneration, we have just scar formation. Let's say the problem is solved, the wound is healed, but it is not completely functional. And this is basically for all types of tissues. What wound healing process normally is the process which, where after the acute injury you have to achieve in, let's say, 100 to maximum 180 days, the full restoring of the wound. First reactions is when let's say, leaving the blood, the blood is leaving the vessel has to be prevented. That is done by the clot. But of course, in this clot, you have thrombocytes, which have also a lot of cytokines and a lot of different molecules, which are also then attracting other cells in our body, which are then working in inflammation process. This one, which I mentioned before. And after this, the cells starting to proliferate, these immune cells and attract also fibroblasts and also other cells which are then starting to divide and also and later produce extracellular matrix, and re-endothelialization is then the end stage, and that is fine. That's for the normal wound. Which cells are involved here? You have mainly, for the cleaning process, you have a lot of metalloproteinases, which are like MMP9 and so, which are clearing the wound. And attracting also macrophages and immune cells, which are then working in the phase which we call wound granulation. And later on, if this is all successful, then the position as an extracellular matrix when collagen is the most normal and also, let's say, in the biggest amounts, but which is also then later forming other cells in the wound and make this reepithelialization, as I mentioned before. If we talk about chronic wound, then after 4 to 8 weeks in the phase of inflammation, this inflammation, the wound is stuck in the inflammation process. And this is so-called chronic wound, which will, let's say, theoretically cannot heal. Because it's, you have to restore the previous situation to really help with the healing. And this is then, let's say, for almost all the tissues, it works like that. It's a model, and this we would like to prevent. So we would like to stop too long and unnecessary inflammation to restore the possibility of regeneration and, let's say, reduction of the scar. And this is now already successfully used in the chronic wound healing processes. So the number of cells here in normal wounds you see, mainly in the beginning they are platelets, then different macrophages, neutrophils, and so on. And later on, when the wound is healed, you have fibroblasts, which are then, because you don't need these fighters, this military, wound military, but you have just, let's say, as I said, extracellular matrix formation, and then wound is fine. So the thing is that very similar is so-called senescence-associated secretory phenotype. This is an, let's say, term which is used for the, let's say, aging tissues or aging environments in our tissue, and which is associated with the, let's say, elderly people, which are a lot of times the result of their different cytokine production and expression is that the cells are stuck in their differentiation. So they, even if they divide, even if they, let's say, grow, they cannot differentiate in the fully functional cells. And mainly you have presence of a lot of inflammatory cytokines like TGF-beta and especially interleukin-6 and so on. And these cytokines are promoting inflammation process and also, let's say, associated problems in the body. This was linked also with, if you remember, with COVID, in the COVID times, with these cytokine storms. It was patients which have very strong reactions. They have similar phenotypes on similar processes in their lungs and in their vessels. So, if we talk about stem cells, what is a stem cell at the end? This is a very big group of cells in different stages of their development, but we can say that we can group them into this cell group if they have so-called asymmetric division. So it means that they, when divide, they reproduce themselves. So it means you get another stem cell, and then in the differentiated cell, which then goes through mitosis. Let's say hepatocyte. It's a liver cell which comes from the stem cell, and then hepatocytes then, let's say, divide and form or muscles, let's say, or other type of cells. Then it means self-renewal is very important. This asymmetric division, so it means that you get two different type of cells after the mitosis, and also which is an plasticity. So it means if you have two stem cells and put one in one environment, let's say in the epithelia, the other one, let's say in let's say adipose tissue, from one the epithelial cell will grow, the other one the adipose cell can grow. But of course, it is important in which stages of development they are. So, and why are so important, let's say, for regeneration? Because they have to, they are like policemen traveling in our bloodstream, and when there is a wound, these platelets and first cells which are coming to the injury are releasing chemokines which are then attracting in this case, so-called mesenchymal, stromal, or stem cells, depends how they call you, but they call them. It's similar, and these cells are then preparing the environment for the regeneration. So that's why they are so important, and they have these trafficking possibilities. That they travel to the places where the injury is, and we are using them also now as a drug delivery systems too, but also for different functions, because they have a lot of different paracrine effects. They are releasing different factors. They are balancing the healing answer and so on. So, but of course, if such a stem cell is then becoming an aging stem cell, it has different manifestations in different parts of the body. Each, let's say, types of tissues have their deposit of, let's say, stem cells, which is then helping the renewal. But it's very, very different because the let's say cells in our body are or organs are let's say they are repairing they are repaired very differently. Let's say heart, neural cells, and so on. Brain very low, epithelia 14 days, and the skeleton 10 years, and so on. So you have very different dynamics and differentiation of these cells. And here you see that also in some cases, let's say you have normally functional potential is lowered, then DNA damage is higher, of course, because these are higher cells. These are older cells. Senescence is bringing, let's say, different expression of the proteins, possibility to make a DNA repair. Also the methylation and different methylation patterns and influence on them, and possibilities how the genes can be activated are let's say in different stem cell aging processes different. So tissues are differently aged. In general, we all know that let's say it's known that telomere, so it's part of the chromosome, and part of the chromosome is like one part of the biological clock. After each division, such a part is, let's say, cut. It's like you will pay for a ticket. And at the end, when it is too short, then also such cells are compromised for proper mitosis. And the result is also that in H stem cells, you have also a lot of these oxygen radicals, which are making damage by itself. And also functionalities. Let's say if you see that in a younger person, when you have a myeloid cell, you have quite a big production of red blood cells, and when there is an aged person, the result is that the number of erythrocytes gets lower, even if you have, let's say, the same number of the stem cells. But also, what is one of the most important part is the dysfunction of the immune system. This is then provoking a lot of other processes, because you cannot balance anymore the proper immune response, and you have this hyperinflammation process then at the end. How the cells then differentiate? It is like let's say, a surface when you have like mountain with different valleys, and if you put marble at the beginning, you know, the cell there, it can go left or right and differentiate at the end, or in fibroblast, smooth muscle, or something else. But normally, let's say, a few decades ago, we said that such cells are not, you cannot make changes in differentiation. So it means you cannot, from one muscle cells, you cannot get, let's say, neural cells afterwards. This paradigm now it's, let's say, changed, so we know that we can make, we can really drive cells here and there and differentiate them, especially if you also know about so-called induced pluripotent stem cells. When you from the skin cell, from the keratinocytes, they can make an embryonic stem cell, which is a so-called pluripotent cells from which all 200 cell types can grow. Most of these processes are done before birth. So these are the transitions, when you have transitions from the pluripotent states to the embryonic, fetal, and neonatal state. So adult phenotype is more or less similar, but we are, let's say, dealing more or less all the time with the adult persons. So what we would like to make, we would like to make from the cells, from the adults, also immortal cells, which can be differentiated in all cell types. Number of stem cells is also declining by age, and you see that this declination comes while you are, let's say, 20, 22 years old. So biologically, you are, the process of getting older is after, let's say, 22 years or 23 years. But first phenotypic reactions, or most of the phenotypic reactions, you see 10 years later, because we all have to know that mainly people, let's say, few hundred thousand years, they were living, let's say, not more than 40 years. And this was also, let's say, when they made experiments on animals, they realized that, let's say, this lower number of the stem cells, if you increase this number by, let's say, multiplying them and then reinjecting them again, you can slower or stop the aging process. And this was done for hematopoietic stem cells. So that was, this was shown that it is quite important. Which cells can be used then for in medicine? Mainly three types, but more or less just tissue stem cells are those which are now, let's say, so-called stem cell therapies today. And you can have them autologous, it means your own, or allogenic, it means from the donor. Mainly are these are hematopoietic stem cells for, let's say, for the treatment of hematological diseases, or mesenchymal stromal stem cells for different anti-inflammatory or regeneration processes. Embryonic stem cells are not used because of ethical reasons. They are just used as stem cell lines. Because in 2006, induced pluripotent stem cells were developed. When you can get, let's say, practically from biological point of view, the same cell, the same type of cell, but without compromising the embryo and let's say isolation of the inner stem cell mass from it. How to then use, where is the future? The future will be in combinations of drugs where we can treat ex vivo or in vivo stem cells to get the proper result. We should start planning more longer clinical trials, because we would like to achieve longer effects, not just, let's say, healing something in the for a short period of time. We will use all the technologies, from nanotechnologies to single cell characterization, artificial intelligence, and new drugs which are achieving, which help then to develop these new stem cells in medicine. Mainly we will work on three fields. One is to get rid of the senescent cells and use so-called senolytic agents. Then, of course, anti-inflammatory drugs together with stem cells, and epigenetic modifiers, which can then, which will be the most tricky ones, which are also linked then with our diet and so on. Just this I wanted to show you. Now I'm at the end of my talk, is how different the cell turnover is when we talk about infants and then aged population, from 5 days to 90 days, let's say, for the same cell type. I have some other slides, but I want to stop here and ask you for the questions.
Thank you. All right, so this.
I would love to hear what GaiaCell is working on right now.
In GaiaCell, we are developing three types. We are working with three types of cells. One is mesenchymal multiplication of allogenic mesenchymal stromal stem cells for treating different also aging, so-called aging frailty. As a hospital exemption, so it means it's according to the legal frame of Europe. Then, of course, for orthopedic applications for different osteoarthritic joints or osteochondral lesions. Our main project is development of CAR T cells for hematological diseases and solid tumors. And the third project is tumor-infiltrating lymphocytes together with enhanced by tumor vaccines to achieve, especially for pancreatic and colon cancer.
Thank you.
Yeah.
I have a question. How do you measure success of stem cell therapy?
That is very good question too, because it is sometimes very personal. Objective, let's say, measurements, let's say if we are talking about aging frailty, they measured a lot of cytokines, but results were so diverse. Just one was showing, let's say, TGF-beta showed if you measure one type of cytokine, you can see if the therapy is, let's say, working well or not. Otherwise, there are a set of different combination of the, let's say, diagnostic measurements, especially inflammatory factors, and also like cognitive, let's say, behavior and so on. If we are talking about
The levels in your blood or it's on the cellular?
In your blood, this is serum measurements. Yes.
I'm wondering if you could talk a little bit more about mesenchymal stem cells, because as you know, there have been a lot of clinical trials, and and no consistent benefits. Is there something about the mesenchymal stem cells that you are making at the company that is going to have quality control that will ensure that they have some kind of function?
Yeah, we have long experience with mesenchymal stem cells from 2014 in clinics when a previous company was asked to prepare for treating graft versus host disease. And we treated 15 patients and it worked perfectly, but mainly for pediatric cases and for acute GVHD. This was one, let's say, application as a hospital exemption. The other ones, let's say, what we saw, but not in the clinical trial, because it's very hard to prepare the objective clinical trial in this respect, especially orthopedic one. It's more or less our scoring, medical scoring to see, and it is very personal. You can use the same cells, let's say, from the same batch with same diagnosis, let's say, same age, but just different patients, and you will get very different results. But let's say, I would say there is a response in approximately 90%, but very good response what we observed, but again with these methods was 20 to 30%. But these data were not published because they are not in the part of the clinical trial, and it's very hard to get these patients back. If it's everything okay, they don't come back. And that's it. And there is no company or no pharmaceutical company which will sponsor such trials. And for a small company, that's impossible. We can make them, we can do it according to the GMP and so on, but that's all.
It's very challenging.
It's very challenging.
And Sonata, the company that you mentioned, the graft versus host, made their mesenchymal stem cells from iPS cells so that they could be more consistent. And I always thought that was a good idea, but I don't know if anyone else has been doing it.
iPS cells are still tricky.
Careful,
very careful.
Wait till you hear my talk.
Yeah. We need something more, let's say. For diagnostics, for this fine, but for therapy, if you have nothing else and you have nothing to lose, then let's say this can be let's say a choice. But otherwise, let's say do it for something simple, I would have doubts.
We'll talk later.
Yeah, of course. Thanks.
Is there time for one more question or not?
Yeah.
Just about the last slide, who was there until 2 seconds ago. I suppose that there are also similar statistics concerning very old people, and I suppose that the rhythm of division is better, let's say, for people living longer than for other people. Do you know more about this? Or I'm curious because I think I never saw, let's say, specific documentation about link between the cell turnover and life expectancy.
No, this combination I never saw, but this, so this is, let's say, I would say more statistical data, and I have to check to the really consistently. I don't know where I got it, but it was interesting. Okay. So more or less, and it's very different if you go, let's say, I have another slide, I'm sorry, but then kick me out. Which I didn't present, but it is here. Oops. Here, so different, let's say, and you know, and then it's very important to know about which cells we are talking about, and I think it's from person to person. And but one thing is, I don't have this reference here, but I'm sure you know it. When they were measuring the number of the clones of the hematopoietic stem cells in the, let's say, children, it was around 25,000 clones, or 26,000. Don't grab me for the exact number. But then there was one lady who donated her body to medicine, and it was two clones. So, and this means something, of course. but let's say if you measure her blood, let's say parameters, they were the same as from the, let's say, from the child. But in reality, it was very close to total exhaustion.
Okay, thank you very much, Dr. Miomir Kneževič.